Okay, perfect. Yeah, so thanks everyone for coming this morning. I'm going to be talking a bit about some of the data that I collected during my PhD, which was actually a couple years ago now, but we were looking at the role of mutual gaze onsets in establishing parent-infant interbrain synchrony. Um, Yeah, and I suppose what kind of differentiates maybe this presentation and this data in particular from some of the others in the kind of series of webinars you may be attending to is like Sophie was mentioning that we have EEG hyperscanning data, but also we also had it, we also collected it from developmental populations. Yeah. So this data is all coming from, two-person EEG hyperscanning recordings from parents and their infants. So, it's definitely a challenging data set to collect, but, super interesting and kind of rich. Perfect. So I'm just going to talk a bit about the background, maybe for some people that aren't from a kind of developmental background too. So it's really revolving around lots of kind of previous research, kind of suggesting that infants are really highly sensitive to mutual gaze. Um, and that mutual gaze is playing a role in supporting kind of early socio-cognitive development. Um, and this kind of thing is commonly evidenced by the fact that infants prefer prototypical eye contact stimuli. For example, things like presented on the screen, like faces with eyes open versus shut, faces showing direct versus averted gaze, and so on. And this type of thing is usually looked at using kind of eye tracking paradigms where the kind of variables are how long infants look at the screen for, or kind of the longest fixation durations and stuff like that. But also evidence in terms of when mutual gaze is disturbed during early social interactions, for example, with mothers that have anxiety and depression. This can also have detrimental outcomes on infants' later cognitive development. Yeah, so plenty of evidence to kind of suggest that mutual gaze is kind of a really important part, and that's not really, uh, very new, a very new idea within developmental research. Uh, but I just want to talk a bit about more kind of specifically the neural evidence, because these are really massive journal articles that have thousands of citations and have been really influential in kind of shaping developmental cognitive neuroscience. So this particular paper by Ferroni and colleagues looked at infants' neural responses measured using EEG to images presented on a screen. These are the images. So one of the two images was presented at a time, either showing a face with direct gaze on the right, yeah, or averted eye contact. And the idea was, in this type of EEG paradigm, you're presented with the stimulus here, it's the face, many times, hundreds to thousands of times, and brain activity is averaged over each time of the presentation, yeah, which is known is on the right, this kind of wave-like structure called an ERP. And typically, what researchers do with this is that they will narrow down a short time window here in the shaded black area, and kind of compare the strength of the signal between these two conditions. Yeah, and that's exactly what Ferroni and colleagues showed, that the infants were showing a more enhanced neural response to the faces showing direct gaze versus averted eye gaze. And this singular piece of evidence has been so fundamental, like I was saying, to developmental cognitive neuroscience, and also to so many theories looking at kind of early socio-cognitive development, like infant sensitivity to kind of communication during social interactions. And I just also wanted to talk about this second piece of evidence, which is also really kind of really influential and kind of within kind of developmental neuroscience. So what this paper did was they were building on Ferroni's paper in kind of a really neat way. Um, the one contrast is that they were looking at oscillatory EEG activity rather than the Ferroni style, which was looking at just time domain kind of changes in amplitude. Here we've got the added frequency dimension. But what they did was very similar type of paradigm where they're showing on a screen either a face with mutual gaze or averted eye gaze. And if you look at the first part, this kind of first box here, and this generally the levels of kind of color here, what they showed is that very similar to the Ferroni paper, that the infant's brains are more activated relative to mutual gaze than non-mutual gaze. How this paper took this finding a step further was they subsequently showed a face with a change in affect. Here, the face starts to smile, and they again presented this with contingent upon mutual gaze or non-mutual gaze. And what they found was that the infant's brains were only really responding to the change in affect in the mutual gaze condition, indicated by this kind of the color here, the red and yellow colors here, and generally the absence of this activation in the averted eye gaze condition. So what this suggests is that not only are infants' brains differentiating between mutual and non-mutual gaze, but actually that the mutual gaze is kind of creating a time period after where infants' brains are subsequently differentially processing information, and perhaps that they're more sensitive to kind of receiving new information. And the kind of why that's important is just to think about generally how kind of communication is facilitated during early social interactions. So generally, these two pieces of evidence are supporting a line of research arguing that infants are incredibly behaviorally and physiologically primed to communicative signaling, and that this plays a important subsequent or follow-up role in supporting communication and learning. But these are all screen-based tasks, and we really don't have the kind of developmental neuroscience looking at correlates of dyadic communication during naturalistic social interactions, which is kind of where the data that I've collected is kind of coming in too. So back when I also wanted to just talk a bit about some of the well, it was recent when I started my PhD a few years ago, but it's really the kind of foundation of the work that I did for my PhD, and that was work by Vicky Leong and colleagues looking at how mutual gaze is processed by both members of the dyad in a kind of more naturalistic and kind of ecologically valid way. And like I was saying, one big idea from that work was, building on the Ferroni papers, was looking at how, during moments of mutual gaze between adults and infants, it's possible to observe increased levels of interbrain synchrony. And like I said, I'm going to be talking about the developmental work that's looked at that by Vicky Leong. But also that at the time when I was doing my PhD, there were a few adult papers that had been published that were kind of showing along similar lines, showing that across entire interactions, you get during moments of mutual gaze, you get increased levels of interbrain synchrony, which at the time felt like a really exciting finding, and definitely something that was really kind of interesting to explore why that was happening. So I'm sure, across the various talks that you've attended, they've all maybe perhaps discussed interbrain synchrony, if that's the measure that they're looking at, in a lot of different ways. And there's some really amazing kind of theoretical papers that talk about the different potential behavioral and kind of neural drivers that could contribute to it. Generally, though, in my presentation, I'm just referring to interbrain synchrony as a kind of, because we use multiple measures, so I'm just referring to any measure that captures um, temporal association between the neural activity of two interacting individuals. Um, and because we go very specifically into the exact kind of chain mechanism that we think that could have been driving the changes in interbrain synchrony, that was kind of, we felt that was sufficient, um, the kind of covering what we meant. So yeah, so this is a specific kind of piece of evidence that was really the foundation of my PhD, and it was performed by both my kind of supervisors through my project. And so what they were looking at was interbrain synchrony between adults and infants. I think they were, I think they were 7 month or just 7 to 8 month old infants here, and the adult was not familiar to them. So it was just a researcher. So in the paradigm, the researcher was singing nursery rhymes continuously throughout a kind of 2 minute, a long section, and they were instructed to kind of either maintain direct gaze with the infant. They also had a indirect condition where both the head and the gaze was averted, and then they also had subsequently a direct oblique, where the head was averted, but the gaze was, directed at the infant. And this was to control for, the variables associated with the head and also the gaze. Yeah. And and they obviously measured EEG from both the infants and the adults. And what they looked at was, interbrain synchrony in the theta band, which is, typically in developmental research, is in the 3 to 6 hertz range, and then alpha activity, which is in the 6 to 9 hertz range. And they found for both theta and alpha activity that they had found increased levels of interbrain synchrony during direct eye gaze condition and the direct oblique eye gaze condition. And what they did with this, with the data analysis, is obviously they had like quite long streams of continuous data, and they, interbrain synchrony was calculated across all time points associated with mutual gaze, and then collapsed down to what you see here, where it's just an average across the entire interaction. So, different focus here, but not necessarily, there's no focus really on kind of fine-grained time dynamics and how that might vary. What it does show us, though, is that, or suggest, is that patterns of brain activity in both individuals are playing a role in how mutual gaze is processed. Independent, kind of additionally to the Ferroni paper or Star paper that was just looking at how an individual brain processed mutual gaze. So like I was saying, these studies don't tell us how increased interbrain synchrony arises around moments of mutual gaze here, because of the style of the analysis where they just average across, the entire time window of mutual gaze. And in order to be looking at, fine-grained changes in interbrain synchrony and perhaps, inferring mechanisms by which, those increases arise, we need to be looking at kind of onsets of behavior. So that's what we designed our data collection around that question and that idea. What we did was, so we had parents, typically mothers and their 10 to 12-month-old infants, kind of interacting across in this tabletop setting. And that was partly due to the kind of constraints of the equipment here. You can see this lead is they're not able to freely move in this kind of EEG setup. What we asked was that the mum would stage a kind of conversation between the infant and this puppet that we gave them, and we asked the mums to kind of shift their attention as regularly as as comfortable between the puppet and the infant. And during piloting, we also had a separate condition where we had toys on the table and we just asked them to play freely. But what we generally found, and the kind of the purpose of this puppet, was that we found a lot more kind of new instances of mutual gaze, yeah, compared to the toy play, which really didn't create loads of new moments of mutual gaze. Yeah. And typically we did sort of 5 to kind of 8 minutes worth of recording for this paradigm. Great. So then what we did was we obviously coded these videos and for gaze behavior, and then we were defining basically mutual and non-mutual gaze onsets, and we also depending on, and we had like different categories depending on what role the infant or the adult was playing. So we defined the sender as the person that was turning to look at the other person, and the receiver was the person that was being turned to look at. Yeah. So for example, as a look to non-mutual would obviously be a time where the infant turns to looking at from the puppet to the mum at a time when the mum is already looking at the infant. That would be mutual gaze, and then times when the infant turned to look at the mum and the mum was looking at the puppet, and that would be looks to non-mutual gaze. Yeah, and in that scenario, the infant would be the sender and the adult would be the receiver. So if you can imagine, if you can imagine kind of, still images of what the infant or the adult would see when they turn to look at the other person, it would, their face and head orientation would look very similar to kind of the Ferroni style images that we, looked at the beginning. And obviously, like I said, we recorded sort of 5 minutes or plus of continuous EEG data. And in order to kind of get this to a really fine-grained temporal level, we pulled out from the video coding these moments of mutual gaze and non-mutual gaze, and we looked specifically at the onset. So time zero in all the plots I'm going to be looking at are onsets. Yeah, and we just extracted very short, kind of 2-second chunks of EEG data around those onsets of gaze. So what we were predicting was that these moments of mutual gaze were going to create basically phasic restructuring of the brain, and because EEG, oscillatory EEG activity has, is obviously, cyclical, and it's possible to measure different phases, and I mean just basically different periods where either it's in a peak or a trough, and that's actually quite, and that's actually the basis of the interbrain synchrony measures that we were looking at. So our hypothesis was that at time zero, which is this black line here, you'd see phasic reorganization in both the mum's brain and the infant's brain at the same time. So such that and each individual line here is a new moment of mutual gaze. So idea is that, both the kind of, both patterns in the mum's brain activity and the kind of oscillations in the infant's brain activity would, become more kind of in line, basically, more aligned following mutual gaze. And that would be important for the measures of interbrain synchrony that we were looking at. So what we did to start with, so now we're looking at the results of this. So the first thing we looked at was these ERP style plots here at an individual brain level. And this was the reason for this is we were interested in building kind of like pathway between the Ferroni style evidence that we talked about at the beginning, all the way up to the kind of Lyon style evidence that we just talked about. So yeah, we first looked at these ERP plots. What we found was that we got really strong brain response, brain changes in relative to sender moments. Yeah, so when either the infant or the adult turned to look at the other person, that created a really strong brain response in the person that was initiating the gaze. Um, and yeah, like you can see here, but what was really not what we expected was that uh, in the receiver's brain, so the person that was being turned to look at, we really didn't observe any changes in kind of brain activity around these onsets. Yeah, and this was certainly not what we were expecting to find. And based on the kind of Ferroni style evidence and the kind of evidence we talked about at the beginning, we were really expecting to also observe really strong brain responses in the kind of receiver's brain activity. Um, the other thing to know about these plots is that we didn't observe differences also here between the mutual gaze and non-mutual gaze. And again, that was, really surprising based on kind of, what we were expecting from the Ferroni evidence that we talked about at the beginning. One thing I would also like to say about these ERP plots is for anyone that's more familiar with EEG research and ERP research in particular, these are not these are not kind of as clean ERPs as you would get typically in a screen-based task. Yeah, here you can see lots of kind of jittery signals, and generally the kind of peaks and the troughs here are less clearly shaped and less clearly defined than screen-based ERP, although the kind of general shapes and structures are very consistent. Um, and what I wanted to talk about, is that we were finding, it was consistently really challenging with this data to kind of interpret what was brain activity, and that's because obviously we were looking at times where the infant or the adult shifted their gaze, so you had a movement of the eyes. And so anyone that knows kind of anything or about EEG artifact and EEG cleaning knows that eye movements and movement generally create really kind of substantial changes in the EEG activity. And obviously we had a situation where the artifact from the eye movement was kind of overlapping a lot in with the kind of brains responding to the kind of new mutual or non-mutual gaze moments. Yeah. So what you can see, this is this frontal image, which is artifact, and then the occipital here is these later parts. It is kind of neural activity or brain activity. So, and you can see that more clearly here in the topo graphs too, where this kind of frontal dark red color is artifact, and then the occipital dark red color later on is brain activity. But there was still a lot of kind of overlap in time. It wasn't just as simple where we could just cut it off cleanly here. And this is really quite we tried a lot of different kind of, the most up-to-date ways that we could think of and we had access to in terms of trying to remove the influence of this artifact and just generally be more confident in what we were interpreting as kind of brain activity. But it was very challenging, and to be to be honest, this is the challenge within all data, and there's not really a kind of clear solution for this just yet. But that being said, we did feel confident about what we were interpreting in terms of actual brain activity. So these next images are looking at that first part of the chain we said, where we're looking at phasic restructuring of the brain activity in the infants and the adults. Yeah, and so this is these are on the left here is the moments where where the infant was the sender, so the infant was the one turning to look at the adult, and the adult was the receiver. And then on the right here is the inverse, where the adult was the sender, the one turning to look at the infant, and the infant was the receiver. So again, a very similar story to the ERPs, where we got really strong brain responses um, in term relative to sender looks to mutual and non-mutual gaze, as you can indicated by these kind of bright bursts of yellow coloring in the sender's brain activity, but not in the receiver's brain activity. Yeah, so this is indicative of kind of changes in phase and phase restructuring. Although one problem with this is that because of how this measure works in EEG activity, when you're in a situation where you have changes in amplitude or power, like we just showed those ERP plots, this can affect this measure. So because yeah, when you have a change in amplitude in the EEG activity, it becomes easier to estimate phase. And so when it's easier to estimate phase, it can mimic, a change in the kind of phasic structuring. So that was definitely the case here in our research, that it was we weren't really able to disentangle the influence of the change in amplitude for the ERP star plots and these changes in phase. So we had some evidence that there might have been phasic changes, but it was difficult to us to say that was, just changes in phase in isolation. Yeah, and again, obviously, it was it was not what we were expecting to observe, in the receiver's brain activity. There was kind of no real response relative to the onset, so time zero, just indicated by the general lack of changes in color around time zero. Yeah. So we next looked at interbrain synchrony. These plots on the top here show time on the x-axis and frequency between 2 and 20 hertz on the y. Yeah, and this is phase locking value, so a measure of kind of phasic alignment between infants and adults' brain activity. And here I'm just showing relative to infant sender, adult receiver, and then adult sender, infant receiver. And generally what you can notice from these plots is the absence of changes throughout the plots generally, and particularly around time zero, there is really no change. Yeah. So, um, yeah. So, and that's definitely, not what we're expecting to observe, based on kind of the theories and kind of research proposed by Leon and colleagues that we talked about before. And this distribution plot it shows kind of chance levels of interbrain synchrony in the theta band, and the red line is how much we observed kind of around time zero. So basically, the amounts of interbrain synchrony we're observing by chance were, very, basically the same as what we're observing in the kind of in our real observations. Which was incredibly surprising to us, and not at all what we were expecting to observe. Yeah, the other thing that was surprising, and again not what we contrary to what we were hypothesizing, was that we didn't observe any differences between mutual and non-mutual gaze in terms of interbrain synchrony. Yeah, there's these plots are just, again, are the same, time by frequency plots. And generally, you can just note the kind of absence of changes in coloration. Yeah. So, one possibility is generally that, our paradigm was the most kind of naturalistic way that this had been done. We used, very like almost entirely free-flowing interactions, and we obviously had data collected from infants that were able to move, and obviously that kind of created generally more noisy data. So yeah, no, it could be that just our measures and our data was not really sensitive enough to detecting these quite subtle effects, maybe. What we would say though is that we were very careful with the data as I hopefully I illustrated before with in terms of our cleaning, but we also we were able to replicate findings. For example, the differences between ERPs to the face versus the puppet in the infant. We found a kind of very well replicated effect there, suggesting that our data was sensitive enough to detecting differences. And I would say generally though that that the mutual gaze findings in terms of the ERP literature are generally incredibly subtle, and they often there's lots of mixed evidence there. So it can be very difficult to kind of pin down what exactly should be observed. However, even the fact that we, didn't show any evidence for changes in interbrain synchrony around onsets of mutual gaze is, still a really interesting point, as what it could suggest is, that in those papers that have shown it are measuring something that's quite different to, what we could observe in ERP style paradigms and ERP studies. Um, And that's because, we found, neural responses that were, in terms of the ERPs, very highly similar to screen-based tasks. So the Ferroni style evidence that we talked about at the beginning. Um, yeah, and I suppose the interesting next step would be, if this isn't something that can be measured in the kind of the way that we designed our paradigm and analysis around looking at moments around time zero and onsets of behavior, then there are other methods that already exist actually that we can measure kind of brain responses in continuous kind of data or kind of behavioral streams, but get kind of a time dimension output to it. So I'm thinking about stuff like regression models and like TRF based temporal response function style analysis, where you'd look at the influence of individual behaviors and how that kind of synchronizes to the data. Yeah. And so another factor maybe is the kind of importance of some kind of temporal regularity in the data. So in this paper that we talked about earlier, where they had found increased interbrain synchrony during mutual gaze, what I didn't mention maybe is that, well, I did, the adult was actually singing nursery rhymes continuously throughout the entire interaction. And so perhaps, we know that infants' brain activity, is capable of synchronizing to singing and speech, and so perhaps generally that these types of external rhythmic influence are creating kind of more rhythmic like brain activity that is and that's easier to kind of measure and observe synchrony in. Although the authors did do a lot of careful analysis to kind of negate the impact of speech kind of on interbrain synchrony. I'm talking about generally the rhythmic structure could create kind of a more conducive environment to measuring interbrain synchrony. Yeah. So then, just to basically summarize, really, we found that mutual gaze onsets were not leading to increases in interbrain, sort of parent-infant interbrain synchrony, which was, really surprising to us based on the Lyon paper findings and kind of the ideas that they proposed in that paper, which, were the basis of my PhD. And yeah, and again, what was really surprising was that our results challenged the idea that infants were really just passively sensitive to a sensitive signals like, mutual gaze, because we really observed no substantial brain responses relative to receiving mutual gaze being turned to look at. And again, this was really surprising based on kind of what the ideas that we thought we knew about from the Ferroni and the kind of Grossman papers that we talked about at the beginning. And yeah, finally, that phasic changes in the sender's brain activity are kind of not enough to drive increases in interbrain synchrony.